Good morning, children. Welcome back to the online classes. We have started with chapter number 5, Principles of Inheritance and Deviations. Today we will start with the topic that is Deviations from Mendelism. All the patterns of inheritance could not be explained exclusively on the basis of Mendel's original principles alone, and certain complexities were observed by later workers which deviated from Mendel's law or Mendelism, and they are, the first one is incomplete dominance. It is a phenomenon in which phenotype of the F1 hybrid offspring do not resemble any of the parent but is an intermediate between the expression of two alleles in their homozygous state. Here, the phenotypic ratio deviates from Mendel's monohybrid ratio, but the parental characters reappear in F2 generation. For example, the dog flower plant and 4 o'clock plant, that is Mirabilis jalapa, the biological name for 4 o'clock plant. Snapdragon or dog flower, Antirrhinum majus is the biological name for it. These are the two types of flower colored in pure state. This is 4 o'clock plant. It is also called partial dominance or semi-dominance. The inheritance in which allele for a specific character is not completely dominant over other allele is called incomplete dominance. On crossing the pure breeding red flower with white flowered plant, hybrids of F1 generations possess pink flowers, and on selfing F1 generation plants, the F2 generation comes as one red, two pink and one white flowered plant. Here, the pink color is due to incomplete dominance of red flower trait over white flower trait. Let us understand the cross. For this, the two parents which were taken are the plants producing red flower and the plants producing white flower. The genotype of red flower is capital R, whereas the genotype of white flower is capital W, capital W. The gametes which are produced by this plant will have capital R as the chromosome, and here it will be the gametes which are produced will have capital W, and here it will have capital R. Now when this gametes fuses, suppose this two gamete fuses, then it results in formation of a new plant having capital R, capital W, which is hybrid, and this plant produce the pink color flower. According to monohybrid cross, the color of the flower should be red, but here the deviation was being observed, wherein the pink color flowers were produced. Now this is what F1 generation, and this F1 generation was obtained by cross pollination. Now when self-pollination of F1 generation plants was done to get F2 generation, then it was found that out of 4 flowers, one flower was red and it was having the genotype capital R. 2 flowers were pink, which was having the genotype capital R, capital W. And one flower was white with the genotype capital W. So, according to this cross, the phenotypic ratio is 1:2:1. Why 1? because one flower was red, two flowers were pink, so two for the pink flowers and again one for the white flower. Okay, and the genotypic ratio is 1 because capital R is for one time. So 1 is to 2. Why? Because capital R W is for 2 times. Right, and again 1 for capital W. So genotypic ratio is 1 is to 2 is to 1 and phenotypic ratio is also 1 is to 2 is to 1. Okay, and in our last video we have studied about Punnett square. Right, so this is the Punnett square with the help of which we can easily find out easily the crosses can be done and the phenotype and the genotype can be obtained. So this is one deviation. Now the second one is codominance. It is the phenomenon in which two alleles express themselves independently when present together in an organism. In other words, it is the phenomenon in which offspring shows resemblance to both the parents. Example, ABO blood grouping. ABO blood groups are controlled by the gene I. The plasma membrane of the red blood cells has sugar polymers that protrude from its surface and the kind of sugar is controlled by the gene. The gene I has 3 alleles, IA, IB and small I. The alleles IA and IB produce a slightly different form of sugar while allele I, that is small I, does not produce any sugar. In humans, each person possesses any 2 of the 3 alleles, I gene alleles. IA and IB are completely dominant over small I. Okay, whereas capital IB and small I are present, then capital IB is dominant on small I, and when IA, that is capital IA and small I are present, then capital IA is completely dominant on small I. So here you can see that there are 2 alleles, capital IA and IB, which are equally dominant. Okay, when they are present with small I, capital IA is dominant on small I and even capital IB is dominant on small I. So both are showing the codominance. Why? Because I does not have any sugar. But when IA and IB are present together, they both express their own types of sugar. This is due to codominance. Therefore, the red blood cells have both A and B types of sugars. Since there are 3 different types of alleles, there can be 6 different combinations. (9q34) is the location or locus of the ABO gene on chromosome 9, which codes for ABO blood group. And total of 6 different genotypes of the human ABO blood types are present as given in this table. You can see here, if the blood group is A blood group is there, then the genotypes can be IA or it can be capital IA small I. If the blood group is B, then the genotype is IB or IB small I. If AB blood group is there, then the genotype is IB. And if the blood group is O, then the genotype is small I. Now crosses of blood group, that is the codominance. If one parent is having blood group A and the other parent is also having blood group A, then the possible genotypes are IAIA or IAIA of another parent. So the phenotype is blood group A, that is of the child. Another possibility is one parent is having IAIA, the other parent is having IA and small I. Then also the possible phenotype of the offspring is A. If one parent is having IA and small I, other parent is also having IA small I, then the possible phenotypes of the offspring is the child may have blood group A or the child may have the blood group O. If the parents have blood group B, if both the parents have blood group B, then the possible phenotypes are IBIB crosses with IBIB, IBIB crosses with IB small I, and IB small I crosses with IB small I. Right? In the first two cases, the blood group of the offspring will be B, but in the last case, there is the possibility of the blood group O also along with B. Now, if both the parents have the blood group AB, okay, then the possible genotypes of the parents will be IAIB and IAIB. In that case, the offspring may have the blood group AB or either A or B. And if the parents are O, have blood group O, okay, then in that case, the genotypes is small I of both the parents, and the offsprings will also have the blood group O. See, this is again if the one parent is having blood group A, other parent is also having blood group A, then the child will have the blood group A or O. Similarly, it is being listed, you can see here. Now we will learn about multiple alleles. When more than two alternative forms, alleles of a gene occupying the same locus on a chromosome in a population, this is known as multiple alleles. The ABO blood group is a good example of multiple alleles. In this case, more than two alleles, that is, three alleles are present governing the same character. Multiple alleles can be found only when population studies are done. Now let us study about pleiotropy. It is the phenomenon in which a single gene product may produce multiple or more than one phenotypic effect. For example, phenylketonuria, a disorder which is caused by mutation in the gene coding for the enzyme phenylalanine hydroxylase. Phenylalanine hydroxylase. The affected individuals show hair and skin pigmentation and mental retardation. Second example, in garden pea, starch synthesis, size of starch grains and the shape of the seeds is controlled by one gene. The genotypes and phenotypes for the traits are capital B, that is round seeds, and the large starch grains. Then the another phenotype is capital B small b, which shows round seeds and intermediate starch grains. Whereas the last one is small b, which shows wrinkled seeds and small starch grains. Therefore, it can be said that dominance is not an autonomous feature of the gene or its product, but it depends on the production of a particular phenotype from the gene product. Now the next one is polygenic inheritance. It was given by Galton in the year 1833, and in this the traits are controlled by 3 or more genes. That is, multiple genes control the traits, and these traits are called polygenic traits. The phenotype shows participation of each allele and is influenced by the environment and is called quantitative inheritance, as the character or the phenotype can be quantified. For example, human skin. Color, which is caused by pigment melanin. The quantity of melanin is due to 3 pairs of polygenes, that is A, B and C. If a black or very dark, then capital A, this will not come, capital B, capital C. And white or very light, then small a, small b, small c individuals marry each other. The offspring shows intermediate color, also called mulatto, where the offsprings have genotype capital A, capital B, capital C. I am extremely sorry, it will be capital A, capital B small a, capital C small a. A total of 8 allele combinations are possible in the gametes, forming 27 distinct genotypes. Now let us study about the complementary genes. It is a phenomenon when 2 pairs of non-allelic dominant genes which interact to produce only one phenotypic trait, but neither of them, if present alone, produces the trait in the absence of other. It shows the phenotypic ratio 9:7. Example, production of flower color in sweet pea. Another one is the epistatic gene or inhibitory gene. It is the interaction between 2 non-allelic genes. Non-allelic genes means alleles at different positions of chromosome loci but can affect one gene over the other in different way of interaction. So it is the interaction between two non-allelic genes. Non-allelic genes, in which one gene masks or suppresses the expression of other. The gene which got expressed is called the hypostatic factor, and the suppressor gene is called epistatic factor. Such an interaction is called epistasis. The epistasis may be of two types. Number one is dominant epistasis. In this, out of two pairs of genes, the dominant one masks the expression of other gene pair. The ratio obtained in this may be 12:3:1 or 13:3. Example is coat color gene in dog. And the second one is recessive epistasis. In this, out of two pairs of genes, the recessive epistatic gene masks the activity of dominant gene of the other gene locus. The ratio obtained in this may be 9:3:4. Example is the coat color gene in mice. Okay. So children, that is all for today. In our next video, we will be learning about the rediscovery of Mendel's law. I hope you have understood this. Thank you.